Hello, good morning class. Assalamu alaikum. Can you hear my voice? Yes, hello. Please let me know if the screen and the voice both are clear. Okay, Nabiha. What about others? Yes, Lujan, Ariba. Everyone, please let me know if the voice is clear. And Lujan, please turn off your mic. Okay. Okay, let's start our class. We are today concluding the chapter Fungi and Algae. This is the last topic of this chapter. After the explanation, we will go through the classwork today. Okay, the written work. So, this is our chapter number 25. Yes, somebody saying something? Yes. Okay, Lujan. Yes. Okay, beta, now please concentrate on the screen. This is our lecture number 25, and we have in the last class we have learned about the structure of algae as oxygen producer, different types of colored pigments in the algae. Okay, today we are going to learn about the structure of Chlamydomonas. I'm repeating again clearly. Please, all of you uh, revise it or pronounce it Chlamydomonas, Chlamydomonas, and Spirogyra. These are two different types or examples of algae. Okay, we will learn their structure in the next slides today. So now the characters or characteristics of Chlamydomonas. Here is the picture of Chlamydomonas. This figure is also given in your book, and on page number 89. If you want to open the book, you can, but right now please focus on the screen. It is a unicellular eukaryotic. We all know that. What is eukaryotic? Eukaryotic organisms are those which have prominent nucleus in their cell. Prominent nuclear structure or nucleus in the true nucleus in the structure. And it is a unicellular eukaryotic green microscopic plant. Since this is a plant, that's why it has chloroplast, and it carries out photosynthesis, means the production or synthesis of starch or food in them. Since we learned that some types of algae, they are also edible, and they are the oxygen producer because they carry out photosynthesis, and oxygen is the waste product of photosynthesis that liberates after the production of food or after photosynthesis. So next character is that it is spherical, slightly elongated. Elongated means ये थोड़ा सा X shape होगा, ठीक है? Elongated, right? Or pear shape. All of us have eaten pear. It's a fruit. The shape of the organism is slightly elongated or pear shaped, right? This is the pear shape or slightly elongated. And this part is the interior part of the organism body. Okay, this part is called the interior means, it is written the spelling of interior ANTERIOR. Interior means the part of the body which is nearer to the head or the front part of the body. Okay, in biology the term anterior and posterior, these are refer to the organism. Interior means the front part or the part of the body which is near the head, like this portion. This is near the head. This is the head portion, and it is called the interior portion or interior part or interior end of the organism body. Okay, so spelling of the interior is written over here. It's anterior end. Anterior end is pointed. This pointed end, ये देखिए, ये थोड़ा सा bulging out है. It is bulging out from the interior end. This is called apical papilla. It is the spelling of the apical, APICAL, apical papilla, PAPILLA, apical papilla. This portion is called the apical papilla, which has a pair of flagella. These are flagella. The single is called flagella and pair is called flagella. What is the purpose of flagella? It is pulsating. Have all of you have seen the cockroaches, and cockroaches they also have flagella. When they move, they keep on moving their flagella. They also have flagella towards their mouth in at their interior end, and with the help of flagella they actually they locate the surrounding situation, or they try to find out what is present in front of them. So flagellum not only help to move, but it also helps to it pulsate means कि सारा टाइम पल्सेस आ रही होती हैं इसमें, पल्सेशन हो रही होती है, लिटिल बिट मूवमेंट होती है, which helps the organism to find out which what are the things or objects are present in front of it. It helps in the locomotion or in the movement as well. Okay, and outer cell wall is present. Look, this is the outer cell wall. This cell wall is made up, please write down somewhere, cell wall is made up of cellulose and pectin. The cell wall of the Chlamydomonas, I'm writing the spelling here in the chat box. Please write down cell wall of the Chlamydomonas. HKL HLAMYDOMONAS is made up of cellulose and pectin. Okay, please note it down. What are the two things which are what are the two substances which are used to made up to make the cell wall of the Chlamydomonas? Cell wall of the Chlamydomonas is made up of two substances, cellulose and pectin. Right? Please take note of it. It is given in your book as well. So this elongated or pointed end of the upper region is called the apical papilla, and it has a pair of flagella coming out, and this flagella helps the or it regulates the movement of water content inside it. Okay, so it helps in not only in the locomotion or to locate the object in front of it, but it also helps to regulate the content of water present in the surrounding. Okay, outer cell wall is present. This is the outer cell wall. It is present and it is made up of cellulose and pectin. Next is a cup-shaped chloroplast is present, which helps in the photosynthesis. Look, this is the cup-shaped chloroplast. Okay, this is the cup-shaped chloroplast is present in the cytoplasm. This entire body is filled with this jelly-like cytoplasm. And this Chlamydomonas, it's actually a plant because of the presence of cup-shaped chloroplast, green cup-shaped chloroplast, which helps to photosynthesize, which helps the plant, this plant organism to carry out photosynthesis and to produce starch. Okay, got it? Next is two contractile vacuoles. Here the contractile, these small dots at the interior and near the apical papilla is called contractile vacuole. Contractile, CONTRACTILE, contractile vacuole. Okay, contractile vacuole is helped to these are present to remove waste matter and excess of water. Who is doing this, by the way? Are you in your senses? Immediately rub it off. Okay, so two contractile vacuoles are present which remove waste matter and excess of water. Means if there is excess water present inside the plant body or inside the body of the organism, it is removed. The excess water is removed by the help of this contractile vacuole, and waste matter, whatever the metabolism or the chemical reactions are taking place, whatever the waste products are produced after a chemical reaction, these are all removed by the help of contractile vacuole. Okay, we have learned in class 7 in chapter cells that in plants vacuoles are present in plant cell vacuole. Large vacuoles are present to store the food. Vacuole is also present to food vacuoles are present to store the food. Contractile vacuoles are present in some organism to remove the excess excess water or waste matter which is being produced after the different chemical activities that are going on in the body of the organism. Okay, so here in this picture they have enlarged it and shown that this is the cup shape. Here this portion have been enlarged and they have shown in this picture that cup shaped chloroplast carrying out photosynthesis, and in the center the red color prominent nucleus is present to control the activities of the organism or the cell, to control the activities, different activities of the cell. This red color nucleus is present in the well defined nucleus is present in the center. After that, the structure which is present, pyrenoid. How do we pronounce it? P-Y-R-E, pyrenoid. Pyrenoid is present inside the chloroplast. This is the pyrenoid structure. Okay, this is the pyrenoid, and its function is to help in photosynthesis, to produce starch. Okay, see it is written, it helps in the starch synthesis. Means when photosynthesis is carrying out, starch is formed after the photosynthesis. The photosynthesis is the chemical reaction when it is carried out in a plant. In all the plants, the food that is produced in the form of starch, carbohydrates, starch. This is called pyrenoid. Here they have shown it clearly. Here they have shown it clearly that pyrenoid is present in the cup-shaped chloroplast to help in the starch synthesis. Okay, right? Next is a light sensitive reddish eye spot. Light sensitive means when there is this eye spot stigma. This is called stigma. This is reddish in color, and this is called the eye spot. What is its function? It helps to distinguish between bright light and shade. Means bright light or dark light. Okay, how the organism distinguish between the bright light or shade or dark by the help of this eye. Okay, when please also remember that when it is asked that in a question, for instance in exams it comes name the organism which has cup shaped chloroplast in its cell. So you should name Chlamydomonas. What is the function of an eye spot? What is the function of pyrenoid? These are very different or peculiar properties of the Chlamydomonas or the algae. These eye spot or pyrenoid, they are also present in Spirogyra, but little bit at the different locations, okay, or in the different form. Yes, yes, Ishal. Yes, Ishal. Keep your copies ready. Let me finish my explanation, then we will go through the written work. Okay. Okay, now next is a big and spherical nucleus controls the entire function of the cell. A big and spherical nucleus is present. I have already told you that a well-defined nucleus is present to control all the activities or function of the cell. Now next is Chlamydomonas. One thing more I want to tell you. Today first we will do question 4, that is in the point form about the structure of algae. I will dictate it. I will show you on the screen as well the written form, in the written form. Then afterwards we will continue with the diagram of Chlamydomonas. You have to draw the diagram at home in your copies on blank page, and I want this green, very neat, tidy, colorful diagram. As you can see here in this picture, in your book, the diagram which is drawn is colorless, but all the structures are very clearly present. You can take help from the this lecture take help from this lecture, or you can search the picture from the Google as well. So please, I want each and every student to draw very neat and tidy diagram. This diagram is very important for your final term exams, and uh, I need very colorful. See the shades, light shades, dark shades, red color, somewhere blue, green color. So all of you should show your uh, like your you should color the diagram in a way that it should look presentable. Okay, next is, this picture has been taken under the electron microscope. In this picture, they have shown the structure of Chlamydomonas. There are so many Chlamydomonas. These organisms are present, and somewhere the dark shade in the cell, it is shown the cup-shaped chloroplast. And inside the cup-shaped chloroplast, somewhere the pyrenoids are also prominent. See, look in this, the cup-shaped chloroplast and the pyrenoids. Okay, right? Next is, this picture is also taken under the microscope. They are showing the activity of flagellum, that oval shape or elongated, little bit elongated structure of Chlamydomonas, along with the flagellum. I will show you the video afterwards when we complete this explanation. Here, this picture has been taken under the electron microscope, and in it they are showing this cross steric section. This is the contractile. This portion is the contractile vacuole, and this is the pyrenoid. And here, this bulging structure is the nucleus. Okay, pyrenoid. Again, I am repeating. Pyrenoid is its function. It is present in the organism to help in the photosynthesis or starch synthesis. Okay, photosynthesis means the synthesis of a starch. Okay, and contractile vacuoles are present to remove the excess water and waste products. Okay, now Spirogyra. Spirogyra, these are actually sargassum. Okay, they are filamentous green alga of the genus that includes blanket weeds. You must have seen on the top layer of the oceanic water or standing water or stagnant water, the dark green color algae. We have commonly call algae. We call them commonly that algae is there, but actually which type of algae, or the example or the name of that particular algae is Spirogyra. Its name is Spirogyra. It's due to the spiral shaped chloroplast. The name is given due to these spiral shaped chloroplasts, and actually they are ribbon shaped. The structure of the Spirogyra, they are, though they are unicellular, but they always form colony. As I have already explained you, that colony means so many unicellular organism, when they live together, uh, they are called their colony. Okay, when so many different unicellular organisms of the same types, they live together, usually they live together, this is called their colony. And uh, Spirogyra, if we look their structure under the electron microscope, they are filamentous. Okay, filamentous means, Huria, your copy is available. It is checked. It is checked. It actually it was kept with me. They did not. The teachers who were providing the copies, handing over the copies, they don't know about it. So I handed them yesterday and asked them, if your father will come again, this copy will be provided to him. Okay, please try to arrange, get your copy from the school. And by the time all those students, those who haven't get their copies back from the school, please, today's work will be done in some rough copy or on some pages, and later on, yes, you have got the physics and chemistry copies are not given. They will be returned back on Saturday. Okay, then your copy is also available on the reception. It's your turn to get it from the school. Okay. Today, if you don't have the all those students, those who do not have their bio copies with them, please take write down the questions today's work on pages, and then paste those pages. Do very neat and tidy work and paste these pages in your bio copies later on. And all those who have their bio copies, please keep them with you. I'm just going to finish the lesson explanation, and then we will move to the written one. Okay, now let's finish the explanation of the spirogyra. Spirogyra, they are filamentous. Filaments means they are filamentous body, and they have ribbon shaped chloroplast, means there is no if no interior or posterior structure in them. There is no differentiation of interior or posterior structure, only they are present in the ribbon like structure or ribbon shape. They are ribbon shape, or some time they people call them in spiral shape. Okay, spiral binding हम करवाते हैं अपनी copies की, they all somehow like that. This is the reason they are named as spirogyra. Okay, due to the spiral shape or ribbon shape. Cytoplasmic strands are present. Cytoplasm are green. These are present. Nucleus is present in the center to control the activities. Pyrenoid are present here to carry out photosynthesis. And again, the cytoplasm are present. Jelly-like cytoplasm are present. Okay, so cell wall, again, it is made up of pectin and cellulose. Chloroplast is present in the ribbon-shaped spiral structure, and nucleus is present. These spirogyra, they occur in the colonial form, in the form of colony. However, they are present in the filamentous form. See, this is the colony. If we look under the electron microscope, we can find a single structure like this. This structure has been taken under the electron microscope. I will be showing you some pictures from the internet right after the class. Okay, this picture is taken. This picture is taken from the colony of the spirogyra. They are attached on the stone or rock. Acha, whatever, wherever they are, these algae, they are present or they are grown, the object on which they are grown, it is called substratum. What do we call it? Substratum. जहाँ पे वो उगे हुए होते हैं या चिपके हुए होते हैं, this is called the substratum. Okay, you can see this is the thick blanket like seaweeds, and they are in the form of colony. Here in this picture, they have shown us the single filament of the spirogyra under the electron microscope. Okay, here there are several different filaments which are present in a thick blanket, and have shown us the filaments of spirogyra under the electron microscope. Now, few questions. Let me quickly finish. We will finish this work. Beta, one thing more. My session will be ended just after 8 minutes. Means I have to reconnect it. So please don't go. Wait, and I will connect it, and we will continue with our work. Okay. Let me show you the. Here, can you see this? Write down in the chat box if you can see this. If it is clear, yes. So type of green freshwater algae, spirogyra, is so named after the spirally arranged chloroplast in its cell. Okay, there are so many pictures which are like this. They have so many from this structure, from this surface of the water, they have collected the spirogyra, and they are in this form. At times people eat them, in some countries people eat them for some medicinal purposes. Okay, and another look, thick blanket of spirogyra. When we right now we can't see that they are filamentous. We can't believe that they are filamentous. But once we see them under the microscope, we can find this type of a structure. This is structure, or this is structure, right? Understood? Another. Uh, okay, just a minute. Okay, Chlamydomonas. If you want to draw the diagram, if you want to see the colors, this picture is very excellent. It's excellent picture to draw a very clear and colorful diagram. You can take help from the Google, okay, for drawing the diagram and for coloring, especially for coloring. Okay, understood? Now please open your copies. Open your copies. I'm going to share the written work with you. Open your copies, your bio copies. Write down the index. I'm going to, after 2 minutes, I'm going to unmute your camera so that you people will, yes. Question number 4, yes. Question 3 you have done on Monday in homework, and today we are going to do question number 4. The index is written first. Write down the index. Without index, I'm not going to check any work. Who is who is this? Who is this? Saim Samad. Okay, quickly write down the index. Today's date. Today is 13/01/21. Okay. Have you written the index, beta, all of you? I'm going to unmute the camera of Abdul Ahad. Abdul Ahad, start your screen, start your video, and show me your work. Rafe, you also. Abdul Rahman. Abdul Rahman and Shahira, I believe that both are sitting together. Please show me your work. Nabiha, you also. Show me your indexes. Yes. Abdul Rahman, show me the index. Yes, Abdul Ahad, good. Yes. Who is this? Fatma. Fatma, beta, isko सीधा करें. Turn, rotate your copy. Yes. Right, right. Good. Abdul Rahman and Shahira. Yes, right. Okay, good. Saim, what about you? Saim. Iman Asif. Faliha, now you show me your show me the index. Yes, good. Abdul Rahman, Maida, excellent beta. Faliha, Zimal, Hamza Mushtuba. Yes, Hamza, I can't see the screen. Yes. Please bring the camera near to your copy. Yes. Good, 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 good. Okay. Zimal, good. Okay, Shiza. Shiza, right. Excellent. Lujan. Yes. Okay, now come on the page from where you will be start writing your. Now turn off your cameras, everyone. Stop video. Turn off your cameras. Turn off your cameras. Now come on the page number from where you will start your work. Your question number here on the screen, you can see the question number 4. Write some characteristics of algae. Quickly start writing. Write some characteristics of algae. I believe that everyone can see the screen. I am going to increase the font size so that it's pretty much clear to everyone. Write some characteristics of algae. Then make heading characteristics of algae in bold letters and underline it as well. Okay, characteristics. There should be no spelling mistakes. There should be no spelling mistakes. Characteristics of algae. Written. By the time I'm marking your attendance, please continue your writing work. Continue with your written work, please, everyone. And please, for those who don't have the copies, write down in on some pages or in some rough copies, because we are not going to upload these assignments again, or will dictate you in the next class or in-person classes. Okay, got it? Barda, just I'm going to unmute you. Show me your copy. Yes, Varda. Talha and Varda. Tabassum, you also. Yes, Tabassum. Beta, start your video. Show me your copy. Who is Shahriyar Nasir? This is someone new, and I don't know who is this. Please, Arisha, if this is you, please rename yourself. Mariam, Manahil and Mariam. Laiba, show me your copy, beta. Yes, Arisha, show me your copy if you have written the question. Shiza, you also. Okay, okay, Saim. Yes, Laiba, good. Talha, please start your video. Yes, who is this? Laiba, yes. Abdul Rahman, show me your work. Yes, Arisha, this is right. Do very neat and tidy work. Beta, do very neat and tidy work. Okay. Yes, Manahil. Sure. Mariam, get the copy from the school. Get the copy from the school. Your copy is there in the pile. It is available here in the at the reception. Please arrange to get the copy. Okay, beta. Okay, Abdul Rahman. It's okay, Abdul Rahman. Now mute yourself. Stop video. Anisha, stop video. The work is still shown on the screen. Please try to complete all the points. Okay, now characteristics of algae. Number one, algae are mostly found in water. Algae are mostly found in water. Full stop. Point number two, they are unicellular. They are unicellular to multicellular in structure. They are unicellular to multicellular in structure. Full stop. Point number three, algae are the simplest plant. Algae are the simplest plants having, the spelling of simplest should be written clearly, SIMPLEST. Simplest plants having no stem, comma, roots or leaves. Having no stem, roots or leaves. Next is point number 4. They contain chlorophyll. They contain chlorophyll and other colored pigments. They contain chlorophyll and other colored. The spelling of colored is COLOURED. Colored pigments. Other colored pigments. Full stop. Next point, point number 5. They prepare their THEIR. They prepare PREPARE. Prepare. They prepare their THEIR. Their food with the help of chlorophyll. They prepare their food with the help of chlorophyll. Full stop. On the basis of BASIS, basis of different pigments, on the basis of different pigments, they are classified CLASSIFIED. They are classified into different groups. Into different groups. Full stop. Point number 7, that is the last point. They are source of food. They are source of food for marine animals. Who are marine animals? All those animals which live underwater in ocean. Okay, they are source of food for marine animals. Okay, Zival, good. We are stopping the question till here, question 4. We will continue with question number 5 and diagram tomorrow. If you have time, please try to draw the diagram on blank page for question number 5 in advance. Okay, this diagram, it is given in your book. You can take help from the Google for coloring purpose. Okay, then we will do the structure. I will dictate the structure in the next class. Okay, just 2 minutes are left. I am sharing the question again. Sorry, our presentation again for marking the pages. Please capture the screen quickly. Just 2 minutes are left. Capture the screen. Page number 89. Done. Then page number 89, second paragraph. Page number 89, second paragraph. Please mark the important points. Done. Then page number 90, page number 89 and 90 both under the heading Spirogyra. These questions we will continue on Friday in our next class, in our last class of this week. Okay, now I'm going to conclude this session with Allah Hafiz, and I hope that everyone has written the points of characteristics of algae thoroughly and understood it very well. If you will be having any problem in understanding, we will discuss this and we will continue our written work on Friday. Okay, Allah Hafiz. Thank you. Yes. Last point बता दें, they are the source. Just a minute. Last point क्या था भाई? Just a minute. They are the source of food for marine animals. Yes, they are the source of food for marine animals. Okay. Allah Hafiz.